Hi everyone, this is Mr. Cinti, and today we're going to talk about the cell cycle. And the cell cycle is basically what the cell does when it's growing, and then ultimately when it reaches a large enough size, it's going to need to divide. And so we want to talk about that. Um, let's talk a little bit about, for example, the cell cycle. Let me sort of increase the size here so we can make it full screen. Let's go here. So the cell cycle, let me diagram here for a second. Like say you had an ordinary cell like this. What's it doing in its life as time goes by? Basically it's growing larger, and it's growing larger still. And we'll talk about what this is called. We have some terms for this. But basically it's making protein, it's replicating its organelles, it's at a certain point in the cell cycle of its life, it needs to replicate its DNA, of course, because if the cell's about to divide, it's going to need another copy of DNA for both cells. But then ultimately, it gets really large, and we've talked about this before. It gets so large that ultimately it's going to need to sort of pinch in into two cells, and this is what we call cell division. This is a better picture of this down below here. And so ultimately what's going to happen at a consequence of this is that there's going to be two cells produced, and these are called daughter cells. And these of course are clones or identical copies of the original parent cell. To throw a term on this, this is referred to as asexual reproduction. Asexual. A meaning not sexual. So both daughter cells are the same as the parent cell. And there's a good reason for that. You want cells to be continuous. You want them, for example, if you have a break in your skin, you have a cut, you want cells to be able to repair themselves, you want cells to grow. And so it's important to have continuity. Let's go back over here and say something about the cell cycle. So one cell is going to divide into another, and it's called cell division. And let me show you this. I drew the cell cycle out sort of linear a second ago, but most textbooks will refer to the cell cycle as a circle, and it's a good way to do that because in fact it's called a cycle. And what we mean by a cycle is that if you look at one cell, what it's basically doing as it grows up, it's going along, and then ultimately gets so huge that it divides into two, and then those daughter cells go on and do the same thing. So it is indeed a cycle. So to throw some terms out here so that we can talk about this a little bit more clearly, when the cell's growing, it's referred to as a period of interphase, and as you can see here, interphase is a really long period of time. I can't say exactly how long because it's different for each cell type, but if we said that the cell cycle was 24 hours, in other words, one day, for the cell to go all the way around to make two daughter cells, interphase would be 23 hours of this, so it would be the vast majority. What we're about to talk about in chapter 10, more so than interphase, is this mitotic phase. It's what the cell does to get ready before the cell divides, and that's called mitosis. And then when the cell actually divides into two cells, that's referred to as cytokinesis or cell division. And so let's take a look at this. So the point of this is that when cells are dividing, here you can see a cell dividing, and this is under the light microscope, and it's kind of cool. You can see how the cell's pinching into two. Now, if you're just a single cell, like a unicellular organism, like for example a protist, or even a bacteria, a prokaryotic cell, when the cell divides, and if you're only one cell, it's full-on reproduction. It's like the cell's dividing into two. But if you're multicellular, like we are and plants are, the cells divide in order to grow. They divide in order to repair. And so that's something to take into consideration. And so let me actually talk a little bit about this process right here. I don't know if you recognize this. This is kind of a famous picture. This right here is a female reproductive cell called the egg. And these little cells over here on the side are sperm. If I were to draw them in like this, you might be able to see them a little bit better if I draw them in like this. And here's the egg cell. It's rather large. You can see here the nucleus. What's trying to occur here is the sperm is trying to fertilize the egg. And so I'm going to talk about that just for a second. As I was mentioning before, if you have one cell and it's going to divide into two, both of those daughter cells are going to be identical. And this is what, if you remember, we called that asexual. In other words, not sexual. And this is important. What's going on here? Well, if you have the father, which produces the reproductive cell, the sperm, and if you have a separate entity, like the mother, who produces the egg, when the sperm fertilizes the egg, let me use some color here to sort of emphasize my point. So here's DNA, like there in red. And it's like, well, wait a minute, if the cell divides, it's not going to have enough DNA. So obviously what the cell needs to do is replicate its DNA. We're going to talk about that. And then partition or separate the DNA so that make sure that it's on both sides. And then, hey, look at this, both cells have the DNA and it's exactly the same because it copied itself, and this is great. But in this case, let me illustrate. The father's DNA will do red, and the mother has a different kind of DNA. So the same kind of DNA, but different base sequence. When you recombine, which is basically what sexual reproduction actually means, it's genetic recombination of DNA. So how does that happen? It's like when the membranes fuse to one another, as you can see in this picture here. They fuse together, and it's referred to as the fertilized egg. Because what's happening here is, let me draw the egg. Let me draw sort of the sperm like this, and here's the flagella. What's happening is the father's DNA is coming in here, and the mother's DNA is recombining with it. And so what you have here is the offspring. So the fertilized egg or offspring is called the zygote. I don't know if you've heard of that term before. It's kind of famous. So we all began life as a zygote, a single cell. And it's like, obviously we're not a single cell now. We're made up of trillions of cells. And so what happened to this cell? Well, this cell then, if you were following this conversation, predictably, this cell needs to divide and produce two daughter cells through asexual reproduction. So this is sexual right here. This is sexual, meaning two parents. The zygote is different from the parents. That's important because you don't want offspring to be identical to the parents always. And so sexual reproduction creates variation in the next generation. It's rather critical. So sometimes you want change in the next generation, but then sometimes you want continuity. So what's happening here is these cells will divide through asexual and divide, and you guessed it. This will ultimately become an embryo, and then a fetus, and then ultimately a baby. And so this process here is asexual, and then this is sexual. So I want to clarify that point before we continue. And so cell division is central to the development of a multicellular organism because the fertilized egg or zygote needs to divide in order to grow and produce more cells. And it's also for repair. Like if I can show you this is what I'm getting at when I say repair. Like let me show you this. So say this guy, he looks like a Stuart Hall soccer player, and obviously he was kicked in the shin and he fell and he tore his knee here. Take a look at this. It's quite tragic. And so if you are able to zoom in closely, you'll notice that the cells of the skin were torn, and then you go all the way down and you're like, whoa, look at all these cells. And so what's going to happen is that these cells are going to need to repair themselves. And the way they repair themselves is they need to divide. And so those cells ultimately are going to replicate. And so let me come back here. It's just a little visual of someone hurting their knee. I hope you enjoyed that. All right, let's go back to the slideshow here. And so what one of the issues is, I mentioned this before, let me highlight this so you can really emphasize, so you can see this. Like if here's the cell, one of the things about a cell is that when it's pinching in like this, it really has to allocate. In other words, it needs to separate its DNA. It has a certain amount of DNA, and then the cell duplicates its DNA or replicates its DNA. And that's easy enough to do, but what's more difficult is that it's got to get the DNA on opposite sides of the cell, because when this cell is dividing, can you imagine, like this? This cell would be fine, because as you can see the DNA is on both sides, but it doesn't just randomly diffuse. If it randomly diffused, the cell would be sort of rolling the dice with whether or not that actually happens or not. So the cell has to take caution to make sure that the DNA is pulled apart. And so one of the main events that we'll be talking a lot about is that before the cell actually divides, it develops these protein strings, which are made up of thick filaments of protein called microtubules. And they form what's called the spindle apparatus, or spindle fibers. And basically, these fibers, as you can see here, are pulling the DNA in that direction and in that direction. And once the DNA is pulled apart, then the cell's good to go and divide into two daughter cells. And so it's not easily done. You're like, wow, that's great. It sounds like it's great, but the problem is that the DNA, it's hard to pull like that. And so here's the cell, and one of the things is it's not only hard to pull, but there's a lot of DNA. And so it's a little bit easier in a prokaryote because there's less DNA and it's a single strand. And so it's a little bit easier for a bacteria to sort of keep track of these matters. But in a eukaryote, we have several DNA molecules, and each species has a different amount of strands. And so you can see the DNA shown in this picture as being sort of a cool orange color here, like that, and it's being pulled apart. Now, you're like, well, what's the big deal? Well, I'm telling you, there's a lot of DNA in the nucleus. It's like if you were to pull it all out, it would be approximately 3 meters in length. And that's kind of a lot. And so it's sort of like taking rolls and rolls of toilet paper, and if our whole lab room was just filled with toilet paper, and I ask you to like grab it and drag it up to the art room, a whole floor up, you would grab it and you'd be dragging it up the stairs, and maybe it would tear, or maybe people would like step on it. It would be crazy to move that quantity of DNA. And you're like, well, what's the problem? What's going on? Like, what's happening with this? Well, normally, here's a picture of the transmission electron micrograph. This is a great shot of the nucleus. You can see over here the rough endoplasmic reticulum. You can see the nuclear pores. Here's a big nucleolus over here. Well, the DNA and the proteins that are inside the nucleus were once called, they still are called chromatin. In other words, the colored material inside the nucleus. And basically what you got there is, what colored material? You've got all kinds of strands of DNA, and you got all kinds of protein in here. And so what happens is the fact that it's loose like this allows for DNA to be able to replicate. But so that's a good thing. The fact that it's loose and you could replicate it more easily, you could sort of read it more easily. It's sort of like paper that's not all coiled up in a spool, but rather it's like drawn out. So you can read it. When I say read it, I mean like turn it into RNA, and you can replicate it. It's fabulous. But it's not good for traveling. It's like if your clothes, I don't know if you're a type of person that has their clothes all over their bedroom floor, but if you're going on a trip, like say you're flying somewhere, you want to take your clothes and roll them up into little tight balls and then shove them into suitcases and sort of condense your clothes into a suitcase so that it's easy to travel in smaller packages. Now, it's obviously not a good idea to wear the clothes, but the cell doesn't need to use the DNA when it's all coiled up. And so I want to talk about this sort of loose and then coiled up nature of the DNA. So I'm going to call this like the good with the bad. I'm going to say that it's good that the DNA is loose in the nucleus, called chromatin. Because why? It's good to replicate DNA that way, because obviously the cell needs to replicate its DNA before it divides. It's good to make RNA, which is called transcription. But the bad part is that the DNA is not easy to travel. And you're like, well, most of the time you don't want it to travel. Most of the time you're not traveling. So it's good that it's loose. So when do you think you should condense your DNA and wrap it up? Right before, you guessed it, right before the cell divides. That's when you want to condense your DNA so that it can travel. And so the way it does it, it's rather clever. I was making an analogy a second ago about like toilet paper wrapping around spools. Remarkably, this is what the cell does. This blue strand right here is DNA, and what these brown things are proteins. They're called histone proteins, histones. And you might guess this: if a protein is in the nucleus, it's made on a free ribosome, and then it simply diffuses through the nuclear pore, goes to the nucleus. And so basically what happens is when the cell is getting ready to divide, it takes its DNA. In other words, you're getting ready to go on a trip. It takes its clothes and shoves it into the suitcase, or in this case, it takes its DNA and wraps it up around these histone proteins. And so it winds them up, coils them up over and over and over and over, and that simply condenses it. I mean, these are incredible pictures here. This is an actual photograph of DNA, that black line right there, in some of our most powerful microscopes. What you see here in the second picture is, let me go back here, sorry about that. You could see a straight line, but then also little spots. Those little spots are the proteins, histone, and the DNA is wrapping around them. So it's starting to coil. Sorry. So it coils further and further and further until it gets really, really thick. It sort of coils on itself. And so let me actually come off of this for a second and show you what I think is a really awesome animation of this coiling process. And so in this case, the DNA is shown here as these purple lines right here, and that green structure is the histone protein. So let me really enlarge it. So what you want to do is coil yourself around those histone proteins. And so this is an incredible animation showing that. Let me show you that again. So here's the DNA coiling around it. And to give you sort of a bigger perspective of this, let's show you this animation. This is pretty cool too. Here, now we're inside the cell. Here's the nucleus, obviously. Here's the nuclear pores. Let's go in the nuclear pore and see what's happening in there. So if we go in, we see all these strands of DNA. And if you remember, it's good. Lots of strands of DNA, and so the DNA can replicate itself. And obviously replication is going to take place during interphase, because that is before the cell divides. And so let's check it out. So if you have a strand of DNA, let's go really close. Here's the DNA, and if it's going to replicate itself, it needs to sort of pinch open. We'll talk about that a little bit more later. So it opens up, and then it replicates into two complete copies. Now these are identical to one another. I just want to pause that and go back just for a second. If you're replicating DNA, the point is that it is actually a copy. So this strand is exactly the same as that strand. And so the point is, if you're following it, when the cell is getting ready to divide, the top strand is going to go to one side of the cell, and the bottom strand is going to go to the other side of the cell. Let me let this play out. But you don't want to just drag the DNA. You've got to coil it up first. So this is what happens. The cell takes its DNA, now shown in blue, and it starts wrapping it around these proteins. I find this to be really cool. Do you notice how these little spikes come off to the side? I don't know if some of you are into knitting, but sometimes these plastic spools contain these little plastic spikes to help hold the yarn a little bit better. And it's curious because the proteins called histones have those same little spikes, which help to condense the DNA. So this upper piece is exactly the same as the lower piece, but check it out. There's so much DNA, and it coils and it coils and it coils, and then it coils on top of itself, and then you're like, whoa, now look at this. Look at what, let me just back that up. Look at this big guy right there. Let me pause it right there. This big structure on the top, this part right here, is called a chromatid. Let me see if I can draw on this. This is called a chromatid. Now, in other words, the chromatin is condensing into a chromatid. I know it's a weird word, but you will get used to it. This is called chromatid also. Oops, sorry. This is a chromatid. Now, because they're identical to one another, sometimes they're referred to as sisters, sister chromatids. I'm not sure if they're sisters of the Sacred Heart, but they're identical, sister chromatids, so they're the same. And so the point is, if you're anticipating, one of these chromatids has got to go to one side of the cell and the other one's got to go to the other side of the cell. But do you see how they're held together here? Let me sort of erase this. They're held together by a central protein called a centromere. And this whole structure, both sisters together, is a very famous, I'm going to get a larger thing, it's a very famous name called a chromosome. So this whole thing is called a chromosome. And so what's interesting is chromosomes are formed in the cell before the cell's about to divide, but each cell doesn't need a chromosome. Each cell just needs a sister chromatid. If you're following this conversation, which I hope you are. Let's go back up here. Go back up to the slideshow. So as you can see here, let's go down here. Notice how when the DNA coils and coils and coils, you have sort of this characteristic X-shaped structure called a chromosome, and each side is a chromatid. And so what's happening is before the cell divides, I mentioned this in the cell cycle, there's a period called mitosis. It's approximately 1 hour. So remember, interphase is really long, like 23 hours. So in a typical day, this would be the last hour of the day, from 11 to 12 midnight. Right before midnight, the cell undergoes mitosis. In other words, the DNA starts to coil around these histone proteins like this, and it forms these thick, short structures called chromosomes. And as you can see, this occurs in the nucleus. And so these are the sister chromatids ultimately. And so they coil around. These are the sisters. They're identical. And then, whoa, look at this. There's so much DNA in a cell. As it turns out, I mentioned this before, that each species has its own number. This is a picture in the nucleus of all of the chromosomes. Look at them right here. It's pretty cool. This is what we look like. We have 46 of these chromosomes. Now, each cell doesn't need one. The idea is that each cell only needs half. In other words, one chromatid is going to go to one side and the other chromatid is going to go to the other side. And so DNA condenses itself for traveling purposes. And so another close-up, here's one chromatid, here's the other chromatid. They're called sister chromatids. And so the DNA replicates during the S phase of interphase, and then chromosomes will start to form during mitosis, which we will talk about in the next lesson. And they're held together by this center protein called, you guessed it, a center part or centromere. And so later these two sisters are pulled apart. Let me sort of conclude with this picture. This is kind of awesome here. This is a picture of the nucleus of the whole cell. All these sort of yellowish fibers are the proteins of the cytoskeleton. So notice how the nucleus is kind of circular and you don't see any DNA, you just see the nucleolus. This would be a typical cell in interphase, 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 interphase. You're like, whoa, what's going on here? What's happening? It looks like there's little orange worms in the nucleus. This is showing you how the chromosomes are starting to form because the cell is getting ready to divide. And so let me sort of leave you with this. The reason for our next conversation is it might be great to have these condensed structures called chromosomes, because you're like, yes, they're great. We'll be able to pull them apart, and the sister chromatids are going to go to the left, to the right. It's fabulous. Here's the problem. There's still a nuclear membrane. So chromosomes aren't going anywhere. And so this needs to be solved. The cell still is in jeopardy, because if the cell, just to emphasize this, if the cell were to divide like that, this cell's in big trouble, this cell's in big trouble. This has twice the amount of DNA, this has no DNA. So the cell's got to solve this. It's step one is condense the DNA into chromosomes, but then you've got to be able to do something. You've got to get rid of the nuclear envelope, and so we're just going to have to wait to see what happens in the next episode of the cell cycle. Thanks for watching.